Hello, everyone. Welcome to the webinar, When Does Aging Begin? Part 3. The webinar is being recorded and will be posted on the GSA website. A notice to all attendees will be distributed once the recording is available. A question and answer session will immediately follow the live presentations. Throughout the program, we will be accepting questions through the questions feature accessible on the GoToWebinar panel. Without further delay, I'll hand the microphone to Dr. Ron Kohansky.
Thank you very much, Judy. Welcome everyone to the third and final in our series on the topic of when does aging begin? Our speakers today are Dr. James DeGregory, University of Colorado, and Dr. Mark Mailer from Einstein College of Medicine. And we will begin with Dr. DeGregory speaking on, in light of evolution, when does aging begin?
All right, so the question we're going to be addressing today is, when does aging begin? And so most people think about this from what would be considered the proximate explanation. So the focus is on damage accumulation, loss of function, increased disease risk, and other age-dependent changes within individuals. So what I'll be talking about is more of an evolution explanation, where the focus is really on the understanding of how selective pressures over millions of years have sculpted patterns of tissue maintenance. And both of these are important, but I believe it's really important that each considers the other. So one thing that my lab likes to think about is cancer. And so we're very interested in the fact that 90% of cancers develop after the age of 50. But we really shouldn't just be limited to cancer when thinking about disease incidence with age, because our physiological decline in post-reproductive years basically greatly increases the risk of all diseases, or many different diseases. And shown here are two examples, which is ischemic heart disease and kidney failure. And what you can see, and I'm going to change my pointer to something really obvious, is that pretty much after midlife, which would be close to 50, there's a dramatic increase, an exponential increase in your risk of these different failures happening. So when this really comes down to a 10,000-foot view from the perspective of evolution, and that there's really been minimal selection against cancer and other diseases of aging past the age where most individuals would already be dead by other causes. So if we look at here an estimated graph of European survival about 15,000 years ago, and this would probably be pretty similar to survival of humans thousands of years ago anywhere on Earth. And you can see that by the time you're 50 or 60, there weren't a whole lot of these individuals around. There were some, but not a lot. And if you look at modern incidence of cancer and heart disease, as just two examples, you can see that there's relatively little overlap between these curves. And thus, from a fitness perspective, cancer and heart disease were not majorly impacting the fitness of these individuals, their ability to reproduce and survive and reproduce and pass their genes on. And so further investment in preventing this physiological decline leading to cancer and heart disease, if that investment meant a reduction of investment in something like reproduction earlier in life, that could have been a wasted investment and would not have been selected for. And this isn't just for humans. We can think about this even when we think about mice. So in our labs, when we study mice, cancers, mostly lymphomas, are a major cause of mouse death. But most of these cancers occur after about 500 days of life, or about a year and a half of life. Rarely do the mice get cancers in the first year and a half of their life. And again, if we look at survival of mice in the wild, you can see deer mice and other mice, basically. Not only is it quite the lucky mouse who lives for a few months, but pretty much it's almost impossible to find a mouse that survives past a year. So again, cancer is not limiting the fitness of these mice. And so when people will often make comparisons between other species like naked mole rats and elephants, and they compare them to mice, one needs to keep in mind, they're comparing to the lab mice, and that's not really a fair comparison. I would actually say mice are damn good at not getting cancer because they don't get cancer to the extent that would otherwise affect their fitness. So for these reasons, life is not linear. And again, I'm just showing you humans, but we could pretty much graph out any species under sort of a captive state, which we find ourselves in modern industrialized nations. And so for about the first 40 or so years, and probably really more like the first 35 years, the odds of dying, particularly say between maturity, age 15, and about age 35, are pretty constant. Your chance of dying in your subsequent year is really quite low, whether you're a 20-year-old or a 30-year-old. And yet we know that that 30-year-old is 10 years closer to the grave on average than a 20-year-old. So even though there's the soma appears to be well maintained, something is happening. And in fact, when people look at parameters of molecular damage, for example, whether it's mutations or other sort of indicators of aging, there's often seen as pretty linear accumulation from maturity through old age. But despite this, our decline is not linear. In fact, it's after the age of 50 or so that not only do we start declining, but we pick up speed. We go downhill faster and faster. And the reason for this delay is really because of resiliency. And I like to think of the 3 R's of resiliency: the redundancy, replacement, and repair. And I'll get back to these later. But basically, these pick up and lift up the curve, essentially helping to maintain tissues to maximize our potential for reproductive success through periods of life when that was likely to happen. We can also see this when we look at marathon world single age records, and that there's basically almost a flat period between about 18 years of age and about 35 years of age, where the world record for any given age is about the same. And again, I know there's complications with this analogy, but suffice it to say, it says that sort of the human potential does have basically a flat period during life. And so this, of course, correlates with the highest odds of reproduction through most of our evolutionary history. And of course, the later ages correlate with the highest interest in aging research. And I've been told jokes fall completely flat over Zoom, but what the heck? So then how does lifespan evolve? And I like to use this analogy from some of Steve Ostad's research. So what he did is he was studying possums from the mainland of Georgia and then from an island that's off the coast of Georgia. And they know geologically that this island's been separated for about 5,000 years. And because of that separation, basically these two populations have been evolving independently. And that's not enough to really have a speciation event, but it's enough for differences to have evolved. And in fact, when they studied these opossums, the opossums on the mainland aged about 50% faster. So by 2 years, they basically had signs of senescence, so senescent fibroblasts in their tails and stuff like that. But the ones on the island aged slower. And so at year 2, they didn't show those same signs. And in fact, they were still producing litters about 50% longer, litters of pups, 50% longer than the ones on the mainland. And so why is that? Well, as you might guess, the ones on the island had less predation. So there were no longer land-based mammalian predators like coyotes and wolves and bobcats. They weren't sustained on the island. And so these animals had the luxury of evolving an extended lifestyle. So basically, they're taking the slow game instead of the fast game. And so this is sort of how you could see this sort of graphically without the cute pictures. So normally under a context of high predation, you could see that the natural lifespan is relatively blunted in this case. And so thus there's strong germline selection for fitness. In this case, we're talking about the fitness of the individual, such that maintenance of tissues and so forth through these ages when there was some decent odds of reproductive success. But then that would decline in old age. And it's not declining because of a program to get rid of the animal. It's declining because of the absence of a program to maintain the animal. But now that those possums are on the island, and now through sustained periods of low predation and less extrinsic hazards, basically, since the odds of survival now have been extended, there's going to be better germline selection for maintenance, and it's going to push this maintenance curve off to the right. And if we again think about cancer incidence, but really you could be thinking about heart disease or anything else, these curves are going to sort of inversely correlate with that cancer incidence. So pushing the curve to the right is going to push the cancer incidence to the right. And so I would argue that if we want to figure out how to reduce cancer and really any disease of aging, we need to figure this out. we need to figure out how are these different species evolved these life extensions, and that, I presumably are also leading to reductions in cancer incidence. So this brings up Peto's paradox. So Richard Peto, I think almost 40, 50 years ago, proposed this paradox, whereas how is it that animals as diverse as like a blue whale down to a mouse, and all these are mammals, similarly avoid cancer through their reproductive periods? And keep in mind that the difference in size of these animals is such that there's probably about a 10 to the 7th fold difference in cell number. And the whales live 50 to 100 times longer than the mouse. And so from the conventional perspective of this sort of stepwise accumulation of oncogenic mutations leading to cancer, this really creates quite the problem for larger animals. And yet they don't have a problem. So how is that? And another thing that we need to consider is, why do men age faster than women? And why do men get cancers at earlier ages? So this just shows sort of a non-scientific graph, just one that we kind of drew up, that represents about this 3 to 5 year difference in cancer incidence for certain cancers that aren't sex dependent and aren't dependent on activities like smoking. And the same thing with survival. There's a similar delay, that women live longer lives than men. So if we think about this, it makes sense from the perspective of, for example, extrinsic hazards and even other hazards, and that males exhibit greater mortality with each year. And this is not only males through historical times, but also males even in modern society. In the sense that with any given year, even in your 20s, a young man is more likely to die than a young woman. It's due to hunting, fighting, risk taking. Males are just bigger risk takers. There's also heavy investment in muscle mass and robustness, which was important because it increased the successful mating, the chances of successful mating, which was better than investing in future longevity. And finally, probably more for females than males, there's inclusive fitness, like grandmother effects, that acts to extend longevity. And there's other complicating factors that I'm not going to be getting into. So let's return to cancer. Again, 90% of cancers develop after the age of 50. So we can think about, why is this? And so largely people think about this from the perspective of, well, you need time, you need cell divisions to create mutations, or you get exposures like through smoking, which can create these mutations. And there's also our underlying genetics that influences cancer incidence. So, in terms of, for example, even inheritance of mutations like in the BRCA1 and BRCA2 gene that increase cancer risk. But really, when people consider all these factors, it all comes down to one parameter: mutations. And this is how the community for decades, really for 70 years, has viewed cancer risks. It's a stepwise accumulation of mutations. But let's step back and think about evolution. So we know that for the last 650 million years or so, there's been this incredible diversity of species on Earth. And this diversity of species has changed dramatically over the ages. Now, is this evolution just driven by the occurrence of the right mutations, over time? And I hope, if you could all speak right now, you'd say dramatically no. Evolution has been driven by environmental change. So these major transitions, evolutionary transitions, have been demarcated by massive changes in environment, sometimes induced by like the meteor that hit off the Yucatan 65 or so million years ago, massive volcanic activity, a snowball Earth where most of the Earth at some period was enveloped in ice, huge changes in gases, big changes in temperature. And these environmental changes thus drove selection for new phenotypes, new traits that were adaptive to the new environment created by these insults. And even within these periods, obviously there was environmental change driving evolutionary change. But it's the environmental change that drives evolution. And of course, mutations are necessary, but mutations are always happening. It's not like these environmental changes are acting as mutagens, they're acting as selectogens. And so we've been thinking about this from the perspective of cancer. And one of the things that we noticed was that if you look at different cancer incidences, you can see that a lot of them are age dependent, and these are for different cancers. And it's important to point out that these different cancers vary by about a log, at least tenfold in terms of the numbers of driver events required to form them. So for example, colorectal cancer is thought to require about 11 different driver events, while some leukemias like CML can be driven by a single event. But if we did a very simple thing, and we just normalized them to their maximums, you could see that these curves very closely fall on top of each other. There are exceptions. So for example, childhood leukemias and somewhat earlier cases of breast cancer in women. But I'll argue that these are either rare or they're due to modern conditions. And thus natural selection has not had the historical ability to prevent them from happening. And so in general, there's a late life incidence for most cancers, as I shared you before. And that has been due to the pressure of natural selection to prevent those cancers from impacting reproductive success. And so one way to think about this, how has all these different cancer incidences been essentially tuned to this late life, even though they're caused by different numbers of driver events, and they even happen in very different stem cell pools. So some work from a postdoctoral fellow that was in my lab, Andre Rosak, what he showed, and he showed this with computational modeling, but I'm going to spare you the computational modeling. But essentially what it comes down to is you can imagine a mutation, that a single mutation like the BCR-ABL translocation in CML that could lead to death. Well, because the odds of that mutation are going to be higher because it's only one required, it should lead to an earlier death of cancer and a lower overall reproductive success. But another cancer might be caused by two mutations, and thus it's going to be further delayed and have less of an effect on reproductive success. Well, natural selection will act such that the red mutation, there will be germline selection for cellular mechanisms suppressing the effect of this mutation, basically the ability of this mutation to lead to a malignancy. And it'll be very strong because the impact on fitness will be very high. And you will also get selection against the blue and yellow mutations, but it'll be lesser so. In essence, it's going to tune these incidences off to the right. And again, there'll be exceptions when, for example, modern conditions are influencing cancer risk, in which case natural selection hasn't had time to catch up. So essentially, we can think about natural selection as being the force that's pushing cancer off to the latter parts of our lifespan. So essentially, natural selection has acted over millions of years to maintain animal bodies to the extent that maximizes reproductive success. And so the first principle is that maintaining tissues in their youthful state is potently tumor suppressive. And the second principle is that aging and tissue damage select for change, which can sometimes lead to cancer. So we can then think about, well, how are tissues maintained in the first place? Because this seems to be still key. So again, I'd mentioned redundancy, repair, and replacement. So we know that, we can have many stem cells, so losing one is not necessarily a big deal. We have repair mechanisms. We have quality control, where a cell that is either damaged or gets an oncogenic mutation can be eliminated. There's immune surveillance. There's cell competition, which is a really cool phenomenon where neighbor cells literally eliminate by either eating or pushing out a misperforming cell that's either oncogenically initiated or less functional. So both less functional and hyperfunctional can lead to the elimination of a cell. And finally, we know that there's peer pressure within tissues. So even a cell that might have an oncogenic mutation, if it's in a normal structure, might basically act normally. And all of these work to maintain this evolved type of cell, which is also essentially the youthful soma. The important thing is all of this is energetically expensive. And so it's going to be tuned to maximize reproductive success, but not more than is needed. And so why does this matter? So because a youthful soma is important not only for as an anti-cancer defense, which my lab focus on, but for disease avoidance, immune defense, tissue function, and really just robustness in general, all of which is increasing the chances that this individual is going to survive and reproduce. And so there's parallels with COVID. Not only do all these other diseases listed over here increase dramatically in late life, the same thing is true for the chance that someone is going to succumb and die from COVID-19 infection. And so we basically have a few take-home messages here. So first is efforts to prevent and treat cancer should converge with similar efforts to prevent other aging-associated diseases. And we need better means to shore up our bodies, the soma, to improve defenses against malignant actors. Finally, I think we need to leverage markers of a declining soma, like clonal hematopoiesis, which I won't have time to go into, as sort of an ecosystem check for our soma. And so when does aging begin? My answer is millions of years ago. So with that, I've already thanked people along the way. Of course, I can't not thank the people that have funded me, NCI, NIA, and Cancer League of Colorado. All the artwork was by my son and lifted from my book. And there's the happy lab all shown together. And thanks so much.
Thank you, James. If there are questions, I can't see any. Judy, could you let me know if I should be seeing any questions?
Not at this time.
Okay, so I will take the host's prerogative of asking a couple. What you sketched out here in this excellent talk was that you have mutation frequency, you have repair efficiency, you have surveillance efficiency, and you have cell competition. The latter three working against the first. So do any of these track with lifespan, or are mutations just geared to mutation frequency working in parallel with lifespan?
Yeah, it's a great question. I don't know of good evidence that, for example, cell competition is it's that, well, actually there is evidence that cell competition goes down late in life. A really nice paper by Liu et al., and I don't remember the details of it. It was in Nature, and they showed that the ability to eliminate a skin stem cell that basically had accumulated damage went down in old age. And that's because essentially the ability to eliminate a less fit cell is dependent on having fit cells around it. And as we get older and more of our cells have accumulated such damage, the odds are it become greater that any given damaged cell will have other crappy cells around it and thus be less able to be eliminated. The other process that definitely goes down in old age is autophagy, which is sort of a cellular maintenance mechanism. And why it goes down is not entirely clear, but autophagy is also a very energetically expensive process, turning over mitochondria, for example. So it seems to decline in older ages. So a lot of, I think, this maintenance pathways does decline. Plus, I wouldn't be surprised if because of this decline, for example, that there's a feed forward loop. So when the intestinal integrity starts declining, now you start getting loss of barrier function. You get microorganism products coming into your system and activation of inflammatory pathways. And this can probably create feed forward loops that then lead to further decline.
So that actually makes a symmetrical statement about how evolution and aging interact, in that you have in the early part of the talk, you showed intrinsic and extrinsic mortality, basically.
Yes.
And now that's at the organism level, and now at the cellular level, there's also cell autonomous or intrinsic regulation, as well as cell extrinsic or cell non-autonomous regulation.
Exactly, it works at all levels, right?
That's astonishing.
Yeah, so essentially, you could imagine that for most animals in the wild, and for most of human history, we were limited by extrinsic mortality. And that's because natural selection had taken care of most of the intrinsic mortality. In some ways, it's amazing. We're an organism composed of 40 trillion cells that we can go for 50 or so years with pretty much a minimal effect. A minimal odds of failure. Not to say that when when disease happens in younger people, which makes it especially tragic, because we're so good at maintaining our systems. And so what humans had to deal with was infections and predation and cold and hunger. Cancer and heart disease were not higher on your list of things to worry about. And same thing within our cells is that, the cells have the intrinsic mechanisms to maintain themselves, like autophagy, DNA repair, and so forth. But those extrinsic controls that basically maintain a tissue are equally important, and that it's sort of a quality control mechanism that allows you to eliminate, sort of a misbehaving or poorly functional cell from your tissue.
Well, it's a great perspective that is going to, I hope, produce some additional research in this area. And I'm happy to say that James is supported by the NIA. On a small personal note, we actually met for the first time at a meeting on cell competition.
Yep.
2 years ago. So thank you very much.
Sure.
Great talk. And now we're going to switch to Mark Mailer on development, epigenetics, plasticity, and aging. Mark, please.
Thank you. I want to express my gratitude to Ron and Judy for the invitation, and to James for his inspiring and really provocative talk. So my talk is going to be based on development, on epigenetics, on plasticity, and on diseases of aging, which are things that I work on and hopefully will help gain some insights into the seminal question of the day. So about 20 years ago, we developed a model of neurodegenerative diseases where we postulated that they were fundamental diseases of development. And here you can see with black that at various stages from gastrulation to adult life, and this is a very simplified version, very simplified version. The black is the pathogenic mutant gene. It has a series of protein partners that are in colors. And at various stages in development, those genes can be activated. And over time, they associate with different protein partners, which allow them to cause cells to become vulnerable, not just at a single focus, which is the focus of primary degeneration, but at secondary and tertiary foci. And B is a second disease, which can start at a different time, and C can start at a different time. And as you can see throughout the figure, you can bring multiple protein partners online at different sites, and these can help promote vulnerability that later gets played out during adult life. Okay. What's very interesting is that cardinal neurodegenerative disease genes, and in this case, presenilin-1, which is involved in early onset Alzheimer's disease, is embedded in a very complex interactive developmental network. And by altering components of the network, developmental alterations can occur, which can impact on vulnerability later. Most of our work has been focused on Huntington disease, which is a neurodegenerative disease that involves motor alterations, cognitive and neuropsychiatric alterations, which start in midlife. And pathogenesis has really been unknown. And we work on Huntington knock-in mice with expanded repeats. And here you can, keeps changing. And here you can see that compared to wild type, that if you look at medium spiny neurons of the striatum, which are the primary sites of degeneration, you can see that at E17.5, at E17.5, there's an alteration in the patterning. The Q18 is the wild type, the Q111 is the mutant. And you can see there's an alteration in the patterning of islet 1 cells, which are the precursors of the medium spiny neurons. And the normal patch and matrix formation of DARP32, which are the mature medium spiny neurons on the top, are dramatically deregulated in H. And so there are developmental alterations that occur. And at the time that we did this in 2009, it still wasn't clear exactly what the basis of that was. The other thing we found, which was very interesting, is that there were alterations in core pluripotency factors, NANOG and SOX2. And as you can see on the top, the expression profiles of NANOG are altered in the Q111. The arrows show that in areas of maturation, there is NANOG present in the mutant. And there's an absence in mature cells of SOX2 in H. In addition, we were able to show that NANOG was deregulated in terms of its phosphorylation profiles. And these suggested that, in fact, there was either loss of cell identity due to dedifferentiation of these differentiating cells, or another process that was involved related to that. We also did BRD birth dating, and basically we were able to show that if you look at the Q111 from embryonic day 12 to 18, there is a very, and I won't use my cursor because I'm going to cause the slides to advance. I can't figure out how not to do that. So if you look at the Q18s, there is a very, very specific profile of tempospatial maturation of medium spiny neurons, which are the red cells that really go from dorsomedial to ventrolateral. And the mutant, particularly at E15.5 in the Q111, that pattern is lost. So the developmental cues for specification and maturation are dramatically deregulated, suggesting that a whole range of cardinal developmental processes are being affected. We then wanted to go on to prove that, in fact, the developmental alterations were related to the actual late onset degenerative disease. And what we did was to selectively express mutant Huntington during development and then turn it off using a recombinant Cree system. And what we're able to show is that the cardinal features of the disease, including striatal degeneration, vulnerability to excitotoxicity, impairments in motor function, alterations in striatal electrophysiology, and plasticity. And these were done with indwelling electrodes to be able to, in awake behaving, freely behaving mice, were dramatically altered, suggesting, in fact, that there is a direct link between the developmental alterations and the late onset disease. We also took embryonic stem cells derived from the knock-in mice and also from knockout mice. And what we were able to show, interestingly enough, is that normal and mutant Huntington were involved in embryonic stem cell self-renewal, cardinal germ layer specification, organogenesis, organ-specific lineage maturation, and also in the process of neural induction and early neurogenesis, suggesting that the vulnerabilities that the pathogenic mutation imparts occurs not just during incipient neural development, but actually occurs at much earlier stages. And this implies that there are vulnerabilities, not just in the brain, but in the body. And we have evidence that these are related to diseases of aging that go well beyond neurodegenerative diseases, as James alluded to. We also used conditional models to selectively ablate normal Huntington in ventral forebrain regions that gave rise to interneurons that were involved in cortical development and neural network formation. And what we know is that in addition to the striatum and median spiny neurons, that cortical interneurons are extremely important in the pathogenesis of Huntington's disease, particularly during critical developmental periods where neural network formation occurs. And here we're able to show by selectively ablating mutant Huntington in regions of the ventral forebrain, in two different regions that gave rise to different types of cortical interneurons, we were able to show that there were alterations in the elaboration of these neuronal subtypes. And we could, again, phenocopy the disease in terms of motor alterations, in terms of pathological alterations, and in terms of behavioral alterations, suggesting, in fact, that the developmental phase of neurodegenerative diseases are due to loss of function effects, suggesting that the developmental component is different from the adult component. The other thing that is clear is that there is a seminal epigenetic scaffolding protein called REST, which normally is involved in repressing neural differentiation genes in non-neuronal cells. And in Huntington, there is an alteration in the nuclear cytoplasmic localization of these cells so that you alter the timing of neurogenesis. What we've done in a large number of studies, as summarized in the slide, are basically to show that REST and some of its cofactors, co-REST, again, which are scaffolds for multiple locus-specific epigenetic factors, are actually involved in the entire process of stem cell-mediated lineage specification of neuronal and glial cells through a range of developmental processes that are pictured on your right. And the mechanisms that regulate REST and co-REST are really quite complex and elegant, and those are pictured on the left. And I'll come back to that because REST is important, we know, in aging, in longevity, in neurodegenerative disease, and so forth, but I'll show another way that it's related. The other thing that's important is to figure out what is the relationship between the early changes and the late onset disease. And this is a group of Hashimoto Tori and Pasco Rakesh that basically showed that if prenatally you expose the developing brain to a series of environmental stressors, you could upregulate heat shock factor 1, which is one of the homeostatic regulators of stress reactions during development. And in fact, what they showed is that if you had deficiency of that, you developed alterations during development, alterations in patterning and specification. But if you had the protective mechanism, you did not see any consequences of the early stressors until late in life. Okay. This is from a commentary article I wrote about one of their papers. And in which they developed on the bottom fluorescent markers that allowed you to trace cells that in red acutely upregulated heat shock protein above a threshold level that induced protection, and in green could follow those cells by using a different system, could follow those cells throughout life and see what in fact was the molecular mechanisms that were mediating the protective effect early and causing it to not be present late. And as you see in the bottom figure above, initially you see in black, you see there is cellular dysfunction, which then gets dampened due to compensatory mechanisms related to these homeostatic systems, and HSF1 is just one of many that are involved. And once these wane, you get dysfunction later in life. And the upper slide basically shows that a whole series of stressors and a whole series of molecular mechanisms impinge on heat shock factor 1, and they activate various regulatory mechanisms in the genome to alter developmental pathways, metabolic pathways, and aging and longevity pathways. And that's important. Two very important articles came out by Dongchang Kai's group from our institution, where he basically showed that microinflammation in the hippocampus can promote systemic aging. Initially, he showed this was due to activation of the inflammasome. And the bottom he showed, which is very interesting, is that it related to hypothalamic stem cells and exosomes and microRNAs that can be transported to the rest of the body and mediate aging effects. And this is very analogous to what has been shown in C. elegans systems from Cynthia Kenyon to Andy Dillon, et cetera. So these studies are very important. The other thing that's important about this is that if you abrogate the inflammation and you abrogate the alterations that you see during development, you can absolutely prevent aging from occurring. Again, suggesting that there's a developmental component that's very important. And here is just, again, a slide which, I thought was important because of the title, but basically it shows that early in life, alterations that occur in the maternal fetal environment can deregulate systems throughout the body. And these are mediated by soluble signals that include neuroendocrine signals, neurotransmitters, neuropeptides, et cetera. And here is just sort of to summarize what was on the last slide, to say that there are multiple homeostatic systems that are involved in brain systemic crosstalk. And at the bottom, you can see, and what I've done is color-coded at different developmental stages what's going on. And what you can see is that there are critical periods, not just during development, but throughout life, that mediate these effects and are constantly under observation. A recent study came out that also showed that the process of aging can be cell autonomous and can occur very early in phase specification. And this is very important to thinking about this and thinking about the process in general. One thing that is very important thinking about all this is reports that have come out over the last few years suggesting that adult injury responses can promote regeneration by causing specific differentiated cells that we thought were terminally differentiated to de-differentiate or transdifferentiate into specific cells that have been ablated, and that these can be involved in mediating regenerative responses, and I'll show why that's important. These processes are very selective. They only affect cells that have been ablated. So major injuries of specific cell types allow other complementary cells to dedifferentiate into the cells that have been lost. And so this process, again, is very context specific. One of the ways of thinking about the entire process of aging, longevity, and is to look at programming. And this is a paper that is in press, is not yet out, basically showing direct reprogramming, where you can basically use certain factors to reprogram an adult cell of one lineage into an adult cell of another lineage, and you can potentially go through an intermediate progenitor state. And what's important about this study is that it shows that you need to, in the process of going to the secondary cell type, which is the neuron, you need to completely obliterate the identity of the starting cell. In this case, through pluripotent factors that act on epigenetic regulators like microRNAs, and that subsequently, to cause the elaboration of the secondary cell type, you use small RNAs that can activate differentiation genes. So that's very important when we talk about cancer in a few minutes. Another study suggesting that, in fact, aging is an epigenetic process that can be reprogrammed is a very important paper that came out showing that by in vivo, giving pulses of essentially the Yamanaka factors, you can recover significant components of aging cells and cause them to become younger. A study that just came out showed that this process occurs in the nervous system, directly involves epigenetic factors like DNA demethylases, and it also suggests that there is an epigenetic memory of the youthful state that is retained even during aging, and that's important. This paper is probably the most important of all, in the sense that true rejuvenation means that within a specific cell type, within a specific tissue, you can take a mature cell and bring it back to an early stem cell. And this is a work by the Piccoli group, where they basically showed that if you supply to either developing cells or adult differentiated cells components, YAP-TAZ, which are components of the hippo developmental mechanotransduction system, you can in fact promote what is true rejuvenation. And that's important in multiple related contexts. We can show that a similar program is involved in tissue regeneration that involves remodeling of the extracellular matrix and is under mechanosensing properties. And so this is a process that is related to rejuvenation, is related to the process of aging and development. And another paper that came out very recently showed that heat shock factor, which can interact genomically with YAP and TAZ, are also important for controlling more than 75% of the heat shock or homeostatic transcriptome, suggesting in fact that heat shock factor, and these hippo-related factors are very important in mediating that process. These things are also important for cancer, and regeneration and wound repair in cancer are linked. So we know during homeostasis, there are stem cell niches that separate the cells on the right that are within a certain niche from those in another niche. During wounding, you can obliterate the niche, and you can cause upregulation of stress transcription factors, which allow a specific program of cell identity to occur by blocking the identity of the alternate lineage. If the stress becomes too great, in the bottom right, you can see that you no longer are able to suppress the second lineage. And in fact, that accelerated stress causes lineage infidelity, and this predisposes to cancer. So these all are, again, potentially developmental processes. We know that the microenvironment is also key to this. And on the right, you show that components of the microenvironment are involved in the regenerative program, and this can be hacked during the process of tumorigenesis. The other thing that's very important is that there are specific cell adhesion molecules which are important for taking a cell that is a precancerous cell and allowing it to become a metastasis-initiating cell, in part by the loss of epithelial contacts and promoting E-cadherin and rest, if you remember back to rest, which allows one to upregulate the cell adhesion molecule to a high state. And this is involved both in regenerative stem cell processes, so wound healing, and also in cancer and beyond that, metastasis, in part by activating the epithelial-mesenchymal transition. Another recent study came out that's very important that showed that, in fact, cancer turns out to be a very high plasticity state, even at early stages. Remember, I showed you in a previous slide that, in fact, when you do direct reprogramming, you need to obliterate the identity of the first cell to get to the second cell. Here, what's very interesting is that in cancer, it turns out, very early on, you have a high plasticity state, and what this does is allow you to express a range of cell types without obliterating other alternate cell types. And so you essentially have a sort of syncytial arrangement that is similar to what you see in pluripotency. And this is what creates a degree of chemoresistance that is very hard to deal with. In a related paper that came out at the same time, these state transitions are mediated by epigenomic processes and are related to RUNX2, which is a factor that is involved in mesenchymal cell elaboration. And mesenchymal cells are part of the process that allow you to establish a metastatic niche. We also know that during tumorigenesis, you can get progressive dedifferentiation of cancer cells to a primordial cell. And this can occur over time and relates to the developmental history of how you originally formed the cell that, in this case, is a lung cancer cell. And so this is, again, redefining an early developmental state, and that can be quite early. And finally, Oliver Holbert's group has been very interested in the relationship between plasticity and cell identity and differentiation. And essentially what he's shown is that as cells differentiate, they activate a terminal selector program, which can activate a large series of genes that are involved in the elaboration of a differentiated cell and also in the maintenance of that cell. And as that occurs, you progressively lose plasticity. And this occurs in large part through epigenetic mechanisms that close off or restrict plasticity. And finally, you can extend this not just to the individual cell, but to the network. And you can show that chemical synapses mediated by gap junctions and connexins in mammalian systems or annexins in C. elegans are involved in this process. So really what we're talking about is that the interplay between plasticity and homeostasis are really crucial for examining all this. So in answer to the question that started all this, I agree with James. I think that aging occurs very early, as I hope I gave some sense of, and is transgenerational, at least from the point of view that I didn't present. So what I'd like to do is just to acknowledge people that have worked on this in our lab and help us think about it, and our funding agencies. And I thank you for your attention, and I'm grateful for the opportunity to speak.
Thank you very much, Mark. Comprehensive. And I think that I'll make one comment, but somewhat in the form of a question, and then I think we need to close. And that is that in the interplay between plasticity and homeostasis, it suggests that the things that cause aging are also opportunities for rejuvenation, and that some aspects are laid down in the epigenome that can be accessed through appropriate interventions. Would you care to comment on that?
Yes, I mean, I think that's very true. I mean, there are 5 slides at the end that I would have made me go over too long, which basically show that there are 30 different processes that have been discovered in the last year or two, which show that everything involved in the chromosome and genome and epigenomic maintenance is laid down during early embryogenesis. So in fact, these processes and factors, which we thought came on much later or took a while to develop, are present at the beginning. And they can be exposed to stressors early, and they can be activated early, and they can maintain epigenetic memories from the earliest stages, although the phenotype may not be obvious. And I think by using multidisciplinary approaches, we can begin to understand what are the epigenetic programs that we can remodel, reprogram, and participate in rejuvenation.
Excellent. Thank you very much. Thank you, James, and thank you, Mark, for excellent and thought-provoking talks. I'll turn it over to Judy for a final word.
Thank you. Yes, I'm sorry we've run out of time. A big thank you to the panel and our attendees. This is the end of the program.